Welcome everybody to today's webinar presented by Biognosys about combining the best of two worlds using absolute quantification of plasma proteins by stable isotope-labelled peptides while doing discovery proteomics using the HRM-MS platform. I'm Florian Marty, Product Support Manager of Biognosys, and I'm your host today's webinar. Later, for the question and answering session, Lucas Reiter, Chief Scientific Officer, will actually join us. The agenda of today's webinar is first an introduction about the single-shot DIA workflow, then how to perform absolute quantification of plasma proteins using PlasmaDive and PlasmaDeepDive, looking into PlasmaDive in action either by doing MRM or PRM. Then we will cover the assumptions to combine the two workflows, looking into the concept that was applied to actually use the results from PlasmaDive and PlasmaDeepDive to do extrapolation for the label-free quantification. And then finally look into the results. First, how PlasmaDive and PlasmaDeepDive can actually be used in Spectronaut. Look into which label-free quantification proxies we were using for PlasmaDive and PlasmaDeepDive, and finally how this actually worked for performing regression and extrapolation to all proteins during the discovery phase. At the end, as I said, we will have a little summary and outlook what's up next, and then have a question and answering session with Lucas Reiter. Generally, proteomics research can be divided into the two topics of either discovery proteomics or targeted proteomics. In discovery proteomics, the questions most often asked is, what happens to my proteome? So what we do is we basically profile proteins across different treatments or different conditions. We are looking into which proteins are regulated, and the regulations are quantified on a relative scale. Using Biognosys technology called hyperreaction monitoring mass spectrometry, with this technology you can quantify up to 7,000 proteins per sample. On the other hand, targeted proteomics asks the question, what happens to my protein of interest? It enables you to quantify a selected amount of proteins with high throughput, determine the quantity of these proteins in your sample, and on top of that perform absolute quantification. And also the absolute quantification can be performed in high throughput. For this, Biognosys offers the technologies called multiplexed multiple reaction monitoring or parallel reaction monitoring, which enables you to quantify around 100 proteins in parallel in one measurement. But why do we actually do just one thing if we can do both? And this is the scope of today's webinar. We try to convince you that it's actually possible by combining stable isotope-labelled standards in HRM measurements to get absolute quantitative information of your spiked-in peptides, while also getting a very accurate label-free quantification of all the proteins and peptides you would discover during your DIA experiment. So for this, we first look into the single-shot DIA workflow, which we term hyperreaction monitoring mass spectrometry. It consists of sample preparation by using either the products such as sample preparation kit that Biognosys offers, together with the HRM or IRT calibration kit, to then generate in shotgun proteomics or data-dependent acquisition so-called spectral libraries, which can be referred as the proteomic inventory of your sample. In the next step, the same sample is then used in data-independent acquisition, and the Spectronaut, our proprietary software, is used to perform targeted data extraction of the data in the spectral library. DIA works in a way that over time an MS1 scan is followed by an MS2 scan, where in contrast to DDA, the selection window of the MS2 scan is rather big. This enables the fragmentation of the entire MS1 range, and no information is lost. Classical DIA is therefore optimally performed in instruments that have both high MS1 resolution and also high MS2 resolution. And as I already mentioned before, it uses the targeted extraction of spectral libraries to deconvolute the data structure of the DIA data. DIA offers several advantages over DDA for discovery proteomics, as can be seen on this slide. So first of all, when comparing the number of MS2 spectra and the number of IDs generated for different gradient lengths, it is actually obvious that DIA identifies more peptides than DDA on all gradient lengths. Furthermore, even on a 2-hour gradient, DIA can identify more MS2 spectra than theoretically possible in DDA. On the other hand, also a very powerful feature of DIA is that it suffers from significantly less missing values compared to DDA, which actually enables much more powerful downstream statistics for these data sets. In the past at Biognosys, we have benchmarked these DIA performances, and as you can see here, first on a setup with 1 meter column, 4 hours gradient, we have identified around 7,400 proteins of a HeLa digest, a single shot measurement, and around 7,500 proteins on a HEC-293 digest, also single shot. Interestingly and very impressively, missing values of only 2.5% with median CVs of around 5%. So basically what you can see is here with single shot DIA, with optimal setup, you can actually identify about 60% of a cell line's proteome with only 2.5 missing values and median CVs of around 5%. Second benchmarking, we have extended the benchmarking from the cell culture onto tissues. Same setup, so 1 meter column for our gradients. In the main difference here was then we have used a resource spectral library that is publicly available. And as you can see here, in one single shot DIA measurement, we have identified over 9,000 proteins with around 10% missing values and median CVs of around 13%. So in summary, single shot DIA enables you to identify around 60% of the tissue proteome, and as you have seen before, also around 60% of a cell line proteome with very few missing values and very low median CVs, as for example here for the tissue around 13%. The second major technology of today's webinar is the absolute quantification of plasma proteins using Biognosys products. Biognosys offers different plasma protein kits, which are called PlasmaDive and PlasmaDeepDive. They are integrated absolute quantification workflows for native plasma, so PlasmaDive, and for depleted plasma, PlasmaDeepDive. PlasmaDive covers 100 proteins in native plasma, whereas PlasmaDeepDive covers around 86 proteins in native plasma and additional 14 in depleted plasma. Both kits are so-called combined workflow kits that combine the sample preparation kit Pro, which can also be bought by Biognosys as an individual product, then the stable isotope-labelled standards, the reference peptides, and the MRM or PRM software Spectronaut. And in combination, the integrated workflow is actually giving you absolute quantities in sample. If you're using the entire workflow together, this is how Spectronaut will in the end display you the results, and you can see here in the marked in red actually the signal quantity, meaning the concentration of your protein in sample. In the past we have tested both kits, so we have tested PlasmaDive and we've tested PlasmaDeepDive using multiple reaction monitoring. As you can see, they are spanning around 6 to 7 orders of magnitude, with the lowest detectable protein in MRM being at around 0.1 nanogram per milliliter. In a more recent application node, we have used PlasmaDive in non-depleted plasma using parallel reaction monitoring. Again, here you can see spanning around 4 to 5 orders of magnitude with median CVs of 5.3% at the protein level for technical triplicates. We've also assessed in the same technical node the lower limit of quantification, median lower limit of quantification using PRM in a multiplexed assay for 100 proteins was smaller than 2 microgram per milliliter, with the lowest protein detected at 43 nanograms per milliliter. Having covered the background information of both technologies, so the discovery proteomics part, the hyperreaction monitoring, and the absolute quantification using PlasmaDive and PlasmaDeepDive, we actually asked ourselves if we can combine the two panels and analyze them in one single run in HRM. Then further, we actually, from our experience, we know that we can identify in total around 300 to 600 proteins in native plasma per run. So the first questions we ask ourselves is, does PlasmaDive and PlasmaDeepDive work in HRM-MS? So can we use the discovery platform to actually generate absolute quantitative information? Then in the next step, we asked ourselves if we can use the generated absolute quantitative information from these 172 proteins to generate a regression and an extrapolation to the remaining proteins that have that don't have stable isotope-labelled spiked-in peptides. Down here is the workflow depicted. So we use, as I said, native plasma, spiked-in PlasmaDive and PlasmaDeepDive, performed single-shot DIA analysis on a Thermo Fisher orbitrap fusion Lummus, performed the targeted data extraction and analysis using Spectronaut 10 spike-in workflow with the DDA library created and the kit file that is sent together with the PlasmaDive workflow panels. So first of all, spike-in plasma was with DeepDive and PlasmaDeepDive was measured in hyperreaction monitoring mass spectrometry. We then determined the absolute quantities with stable isotope-labelled standards. Next, we determined label-free absolute quantity proxies using different methods such as top 3, sum all, or some IBAC corrections. Then we generated a regression of absolute quantities based on the stable isotope-labelled standards and compared them versus our label-free quantity proxies. And then use this regression to estimate the absolute quantity for all proteins measured in HRM-MS, additional to the proteins subjected with PlasmaDive and PlasmaDeepDive, and then also tested on top of that again different LFQ proxies, which is also part of the ongoing work. So first the results. So we have tested PlasmaDive and PlasmaDeepDive in Spectronaut 10 using the spike-in workflow, and as you can see here in this slide, if you're using the PlasmaDive with the spike-in workflow, what you can actually see is an overview of how the data looks. So we have identified in the first round around 6,100 precursors in the label-free workflow, plus 127 precursors from the spike-in kit. And as you can see down there then, if we're looking at the first protein, that you actually identify both precursors and the fragment selection, everything worked. Visualization of the spike-in workflow is pretty similar as in Spectrodive. So you get the target to reference ratio, and in the future we're also thinking of implementing the absolute quantitative information. Here just a closer look up at the same peptide, and you can also see that for some of the proteins where we have stable isotope-labelled standards, we did actually not identify the corresponding light target. So these are most often the proteins that come from the PlasmaDeepDive kit, which would be accessible when using depletion. We have then tested how this analysis correlates. So how can we actually correlate the absolute quantity determined by PlasmaDive and PlasmaDeepDive, how well this correlates with the intensity of the corresponding light peptides. So how accurate actually is the absolute quantitative information we get in the discovery phase. And you can see here an R-square of 0.94, so this worked pretty well. So coming to the first summary, we saw that PlasmaDive and PlasmaDeepDive, they work in hyperreaction monitoring. All 172 stable isotope-labeled peptides were measured in hyperreaction monitoring mass spectrometry. 139 out of the 172 stable isotope-labeled peptides, we actually measured the endogenous peptides. We could generate absolute quantities based on the intensity, and these quantities, they actually correlate well with the absolute quantities determined from the peptides from the kit. And as you have seen here is Spectronaut can actually process these kind of data sets without intensive manual adjustments. In the next step, we then looked at the label-free quantity proxies using PlasmaDive and PlasmaDeepDive. And what we did here is we basically used half of the panel to generate the proxies, and then use the other half of the panel to test it. And in a first set, we got an R-square of 0.7. We then performed some different proxy settings. So we looked at sum of all peptides. We looked at top 3, top 5. We did some IBEC correction. We also used it without IBEC correction. And in the end, came up with the best solution in this case was the sum of all peptides for doing the protein quantification. Then we also did an error estimation, and we came up with an absolute error of around plus minus threefold, which is well in agreement what people are constantly reporting when doing these kind of absolute quantitative proteomics workflows. We then went deeper into the data and tried to understand why we actually got only an R-square of around, for this run, 0.68 or 0.72 as for the other one, and looked into some of the candidates, some of the proteins. So what we could see is on the upper left and circled in green, so we have proteins that are based on the stable isotope-labeled standards. They are overestimated by our label-free proxy. The one in red, they are actually underestimated by the label-free proxy. So we wanted to look into these, what might be the problem. So the first problem that we saw is that for the proteins that are overrepresented by the stable isotope-labeled standards, we are mainly dealing with interference, as you can see here for the Y5+. If we're actually manually correcting this, then we can already see a major improvement in the quantification of these proteins. For the proteins being underestimated by the LFQ proxies, we tested a few things, and in the end came up with the solution that using only proteotypic peptides improve the entire regression. In the third iteration of going through this manual interference correction and also using only a proteotypic peptide, we could actually improve our R-square from 0.68 to 0.81. So what I have shown you so far is that we have generated label-free an LFQ proxy. We have used that proxy to generate a regression, and then have used this regression to perform the extrapolation on all of the proteins identified. As you can see here on this plot in blue, we have the quantities of all the proteins that we have determined using PlasmaDive and PlasmaDeepDive. Then we plotted the average of 3 biological replicates, and also plotted the quantities of all the quantities determined by our extrapolation. In total, in 3 rounds, we have identified around 365 proteins here in this experiment. So what I've shown you today so far is that PlasmaDive and PlasmaDeepDive can readily be used for HRM-MS with Spectronaut. Absolute quantitative information generated here correlates very well. It allows you for reproducible absolute quantification across the whole abundance range of plasma. Workflows are fully supported without the need of extensive manual adjustments. And that the spike in peptides, they can be used for LFQ absolute quantification estimations using the proxy and the regression I have shown you so far. Further, actually also shown that we get very low median CVs for biological replicates, meaning that the entire workflow is very robust. And in summary, we can say that we can combine the advantages of discovery and spiking in absolute quantification And this is especially valid when discussing large scale studies where people want to actually look into hundreds of thousands of samples, where the discovery part becomes even more important. As I've said in the beginning, this is still ongoing work. What we are currently doing is we are looking into refining the LFQ proxies. We are also thinking into take into account features such as peptide flyability or other parameters that could improve the proxy. Then we are looking into more details, how to handle proteins with similar protein sequences, so how to deal with proteotypicity and all this. And then also extending this entire workflow onto a larger scale study where we are analyzing 10, 20 samples to get a better feeling of how accurate and how robust it is. Okay, with this, we're actually coming to the end of today's presentation. And with me here now is Lucas Reiter, who is more than happy to take your questions. Yeah. So the first question is, can you please explain in more detail how you extrapolate the abundance for non-spiked-in peptides?
Okay, of course. Welcome everybody. So the way how this works is that for the peptide and for the proteins we have in the PlasmaDive and PlasmaDeepDive panels, which are a little bit less than 200 proteins, we can determine the protein absolute amounts using the stable isotope standards we have in these panels. So for the stable isotope standards, we know the absolute amounts we spike into the samples, and using that we can calculate approximate absolute amounts for proteins. And then for the same proteins, we can also calculate a so-called label-free absolute quantity using various published methods. This is what we call here the LFQ proxy. So this can be based, for instance, on the top 3 flying peptides of a protein. And then you get a certain value, and this will correlate with the absolute quantity of a protein. And so you will get a pair of data points for all of the proteins in PlasmaDive and PlasmaDeepDive, and you can generate the regression of the protein absolute quantity as determined with the stable isotope standard versus your label-free absolute proxy or your label-free quantity proxy. And using this regression, you can then extrapolate to all other proteins that you have identified in your discovery study. So this is basically the way how this works. This has been published by a number of people, and we just tried it here with HRM and with our PlasmaDive, PlasmaDeepDive panels, and we saw that this works really very well.
Okay, the next question is, how many points across the peak were you getting in the examples that you showed?
We typically aim for in the range of 6 to 7 data points per peak when we define the peak width as being 4 sigmas or peak width at 13% height of the peak. This is what we typically aim for, and the reason why we aim for this number is that we saw in the past that we get the best quantitation if we use methods that produce this amount of data points per peak. So our benchmark criterion is typically the number of peptide precursors we identify with CVs below 20% in technical triplicates. We always aim to maximize this value.
Then the next question is, how are the missing values defined?
I think this refers to the introductory slides. in the numbers that Florian showed here, this refers to protein quantities. Of course, if it depends a bit how you define the missing values, whether it's on the peptide precursor level or whether it's on the protein group level. And the missing values will be slightly lower if you define it for the proteins and a little bit higher for the peptide precursors. It's just a matter of definition, basically. So for proteins, we get very low numbers of missing values in the range of 2 or 3%, what Florian has shown here. This is always for technical replicates of the same sample. If you have biological variability in, then this number will go up a little bit because a few proteins will simply be below the detection limit. Yeah, so it depends on the experimental setup and on the way you define the missing values.
Next question is, can or will this workflow be portable to other types of samples, such as tissue samples? So in theory, this workflow is portable to any kind of tissue, any kind of cell culture. It's also applicable to any kind of panel of stable isotope-labelled standards. And whenever you want to actually implement such a workflow, just send us an email and we help you to set up the workflow in the correct way, also using the right settings within Spectronaut.
I can also add a little bit something to that. In many tissue samples for human, there is also contamination with blood, for instance in liver tissue. And in those kind of tissues, PlasmaDive and PlasmaDeepDive are also readily applicable because you will identify a lot of blood proteins in these tissues as well. And this applies not only to liver, but also to other tissues. So even with PlasmaDive and PlasmaDeepDive, even those two panels are applicable to quite a number of tissues. And as Florian said, you can extend this to any set of stabilized top standards for any tissue with Spectronaut. That's not a problem.
The next question is, could you comment on the use of the Lumos versus the Q-exactive HF, for example, in this workflow? So the entire workflow, as we have demonstrated here, is working on any kind of instrument from any vendor that can generate DA or SWAF data. And in this case, we have simply used the Lumos because it was available in our facility at that time. But you could also do it as easily on the HF, for example, if this is the instrument you have at your availability. Then the next question is, how do you deal with peptides from your absolute quantitative spike-in, which give different values for protein abundance?
I think this question refers to when having multiple stable isotope standards for a single protein. In PlasmaDive and PlasmaDeepDive, the peptides have been very carefully selected, but in the end, only a single peptide per protein was, a single prototypic peptide per protein was selected. And in the selection process, we looked at the reproducibility of quantitation when you do multiple sample preps, the stability of the peptides, and so on and so forth. We did not select the peptides necessarily for how accurate they would quantify a protein in absolute terms, because for this you would need protein standards, and we did not do this kind of selection for PlasmaDive and PlasmaDeepDive.
Next question is, you mentioned that you're working on a large-scale study which is 20 to 30 plasma samples, correct? So I think this was a kind of a misunderstanding. So this is what we are planning on expanding this entire study.
Yeah, so doing DA in plasma can be easily extended to hundreds or even thousands of samples. We have done this in our lab already, and we are planning to do this even more in the future. So this is a very scalable workflow.
Then the next question is, do I understand it that in your final curation you use only the proteotypic peptides in your top 3 LFQ and that this is now the standards? So we actually did the entire LFQ proxy with using some all peptides, but only of the proteotypic peptides. This is by no means what we define standard. It's just what we have done and got the first and best experience in this study, and we're definitely looking into that, and by generating more data sets, seeing what might be better options. Then the next thing is, can you talk a little more about the advantages, disadvantages of using label-free versus with label MS when doing comparative proteomics?
Okay, so I think the major difference between these two approaches is, there are two major differences, I would say. One is that it's easier to compare data sets over a long period of time when you have stable isotope standards available. So you can, in principle, you can set up a assay panel and a sample prep protocol, and you can acquire data over years, and the quantities you get out should be very much comparable, even if you have a change of platform or an update of platform in between. Whereas if you're using label-free quantitation, there are certain limits to changes of the platform because you can only correct so much afterwards in your data processing. Of course, you can do normalization of data and you can do batch correction of data, but there is a certain limit to what you can correct out. And I would say it's definitely a good practice to acquire label-free data on a very similar platform all the time. And if there has to be a change of platform, then the data is not necessarily easily comparable anymore.
Okay, the next two questions, they go into the same direction. It's basically about using the extrapolated values and doing cross-referencing to either literature or other information panels where you actually get the absolute quantitative information. And as Lucas has already said, so we have not done that so far because first of all, as Lucas mentioned, you would need protein standards to actually get protein quantities. And second of all, this is all just work in progress. We just got the results a couple of weeks ago and are now looking into all these questions. How well is it actually correlating with reported stuff? So there's definitely stuff going on, and we will continuously reporting on how this progresses. The next question is, for specific disease, do you recommend adding disease tissue samples into the spectral library? If so, do you need to dilute the tissue digest with plasma to minimize matrix dependent retention time shifts and other things?
Okay, I think I understand this question. So this still refers to plasma, I assume, and it refers to leakage of tissue proteins into plasma. I have to say we do not have so much experience with that. I have had a few projects in contract research that were along these lines, for instance with pathogens or other diseases. Yeah, we've had success doing that, but I think it's very, very experiment dependent. I would say with Spectronaut it's easy enough to just generate spectral libraries from plasma, including the pathogen, and from the pathogen alone, or from the tissue where the pathogen is alone, and build all of these spectral libraries and just check which one of the spectral libraries works best on your data. But I think it's difficult to give a general answer here.
The next question is, the minimum number of peptides that you need to be spiked in for an accurate estimation across a highly complex sample like cell lysates?
It's probably important that sufficient that there are sufficient orders of magnitude covered with the stable isotope standards. And I would say if you are in the range of 4 to 5 orders of magnitude with your stable isotope standards, since it's quite linear, or since it's actually very linear, this should be fine, I would say. But of course, the more data points across the dynamic range you have, the better.
The next question is, can a test for interference be automated to preclude the need for manual examination of thousands of peptides? So the interference correction applied by Spectronaut is per default on and is also automated. And as I've shown here is, actually in this case, we only had to go and look into the peptides, so the one that the stable isotope-labelled peptides of actually 2 proteins. So there's absolutely no need with Spectronaut to go through thousands of peptides, and as I said, the algorithm is very powerful and is removing most of the interference.
I can also add a little bit more technical to that. The interference correction is based on the principle to remove ion traces that do not follow the consensus profile of this peptide, so the consensus solution profile in the chromatography. And in the example Florian has shown, the interference exactly follows the consensus elution profile of this peptide. Now there could be multiple reasons for that. It could be that the interfering peptide elutes exactly the same as the peptide of interest. And in this case, it gets quite difficult to remove an interference automatically, or it's not as easy. The other possibility is that you actually have an overlap of 2 fragment ions, one coming from the heavy peptide and one coming from the light peptide. And this is something one could definitely add additionally to the current interference correction, and this could also be automated. So we will probably look further into that.
Then the next question is, can you use PTM modified stable isotope-labeled peptides in this workflow? And I would say in general yes. I mean, with the known limitations that these PTM modified peptides are actually having. And the next interesting question is if there is a purpose of using the IRT peptides as an LFQ proxy, since they're already included in the sample, with, for example, spiking in known amounts of IRT peptides. And I would say there the main problem is that you only have 11 IRT peptides, so you would have a very limited data set of peptides that can be used for this LFQ proxy.
Also, you cannot make a regression for absolute protein amount to label-free absolute protein amount, because you actually don't have IRT proteins in your sample. So this would not work in the setup we have used here in this study.
Okay. If there are no further questions, I would thank you all again for showing up to this webinar about combining the best of 2 worlds. As I said, we'll follow up on this. We will also keep you in the loop and provide you with more information. And as I said, when you're interested applying the same workflow either with our PlasmaDive and PlasmaDeepDive panel or with your own panels of stable isotope-labeled standards, contact us, either directly me or at support@biognosys.com, and we will provide you with all the necessary information to apply these workflows.
Thank you very much for your attendance. Goodbye, everybody.